Hello everyone and welcome to today's webinar, organised by The Medicine Maker and CRB. I'm Stephanie Sutton, editor of The Medicine Maker, and I'll be the moderator of today's online event, Cell Therapy Facilities, Designing for Flexibility, Scalability and Cost Effectiveness. Our speaker today is Peter Walters, a lead process engineer with CRB. Peter has over 14 years of experience in pharmaceutical process and facility design, including the design, development and validation of pilot and manufacturing processes for aseptic bulk production systems. He has extensive experience with cGMP manufacturing, including the generation of procedures and batch records, validation of systems and processes, and the manufacture of clinical and commercial products. He has designed process facilities for monoclonal antibodies, gene therapies, allogeneic cell therapies, and aseptic novel processes in both stainless steel and single-use systems. He has also helped author a new drug application, participated in FDA facility inspections, and is a co-inventor for a patented novel product formulation process. Recently, he was also awarded the esteemed 40 Under 40 award by Consulting Specifying Engineer magazine for 2018. Peter will be on hand to answer any questions you may have at the end of the presentation, but remember that you can submit questions at any time throughout the webinar by clicking on the Ask a Question box on your screens. So Peter is now ready to start the presentation, so I'll hand over to him.
Hello, welcome. Today I'll be talking about cell therapy facilities, specifically around designing for flexibility, scalability, and cost effectiveness. This webinar will provide an introduction to design issues for cell therapy facilities in general, specifically around the operational spaces and how GMP and biocontainment requirements impact their design. And an important aspect of this is the identification of required GMP support functions. Mechanical and electrical will be considered and their impact as well on the facility. And we'll introduce regulatory requirements and how we can design to them in a cost-effective and flexible manner. A quick overview of the presentation on a whole. We'll be discussing guidance background, what documents we'll be referring to in this presentation, what's available specifically to cell therapy, and then a quick overview, high-level overview of the cell therapy process. And I feel that this will help form a foundation for design implications that we will discuss throughout the presentation. The processing space, we'll talk about how those implications play a part in the design of a facility in your operating space. And then we'll do a discussion of the facility, including support functions and how the facility on the whole is impacted by cell therapy processing. And at the end, we'll talk about a case study with cost considerations will be looked at as well. So I want to open with discussing about PICS, or Pharmaceutical Inspection Cooperation Scheme. It's an assembled committee that works to harmonize GMP guidance for the industry, kind of across the world. Not every country or regulatory body participates in it, but it's currently comprised of over 52 different authorities across the world. So I like to use this as kind of an international basis because it really touches more than just local authorities. It looks at international trends. PICS tends to borrow heavily from UGLEX, the regulatory documents for the European Commission, with various annexes that pertain to specific product or industry function types. The latest release was on January 1st, 2017, so it's fairly up to date in terms of technical knowledge and understanding, and in terms of thinking by those regulatory authorities. And while having a internationally compliant facility is maybe less impacting on cell therapy facilities, as we'll discuss, mostly because autologous or patient-specific products maybe aren't traveling throughout the entire world, so there's really less need for a fully internationally compliant facility. I think it's still very important to look at these kinds of documents because international trends may bleed into the more local regulations, and so understanding the international trend in the way that the harmonized guidance documents are heading can really give you insight into the future of where the regulations are going to be going. And then UDRLEX just released guidelines on ATMPs, or Advanced Therapy Medicinal Products. And this is specific to cell therapies and gene therapies, which we won't be talking about today, but cell therapies, both autologous and allogeneic. And this was adopted November 22nd of last year, so it is fairly recent and, again, pretty up-to-date in terms of where the thinking is. And since historically PICS has borrowed so heavily from the UDRLEX, this is a good document to look at in terms of where world expectations may be going again. And then for FDA guidance, HCTPs, or human cell and tissue products, pretty much fall into two different categories referred to as 361 products or 351 products. And 21 CFR 1271 gives the criteria for that differentiation between 361 products and 351 products. 361 products are things like bone, ligament, cartilage, skin, vascular grafts, things like that. And 351 products are all of those that don't qualify under those differentiations. And so those are going to be things like cultured nerve cells, gene therapy products, human cells used in therapy that involve transfer of genetic material. So we're going to be discussing exclusively 351 products as far as the regulatory understanding goes, which of course would include autologous and allogeneic therapies. So as a high-level overview of cell therapy in general, right, what are we talking about here? There's two main classifications. The first I'll be talking about is allogeneic therapy, and this is where the cells and tissues come from a donor or a donor pool and then are administered to a patient that is different from that donor. And so because harvest of the cells is removed from the patient, it can be done in advance and in anticipation of having to dose a patient. The result is an off-the-shelf opportunity, if you will. And this allows for an available range of scale because you could potentially be producing this product line in bulk or for multiple patients at a time. It is important to note that there is allogeneic donor match products where you are basically matching from a specific donor to a specific patient. So that would be performed more on a patient-specific scale. But if it's non-patient specific, that can be produced in bulk, and so you can get up to larger scale production volumes. And so benefits of an off-the-shelf opportunity for cell therapy would certainly include things like being prepared in advance, which would allow these products to be ready to use at hospitals to immediately administer. It also allows for better risk mitigation because you have a larger supply of product available. So in the case that there's a contamination or an issue with one or two doses, there is a large supply available to reach the patient as opposed to having none left available. And then the other main classification is autologous therapies. And this is where the cells or tissues are harvested from the patient themselves and then readministered to the same patient. So this kind of therapy is going to be exclusively patient-specific scale. The benefits of a patient-specific therapy is that there's going to be no human leukocyte antigen matching needed because the cells are coming from the patient themselves, And so then the patient's body is going to be able to recognize those cells. Likewise, there typically isn't any immunosuppression that's required for administration. And so then the effectiveness of this kind of therapy tends to be increased comparatively because there's reduced rejection of the product line. However, with this comes a number of complications, as you might imagine, specifically around logistics. The patients may be heavily treated by the time that this therapy goes to be used, so the cells that you are harvesting from the patient may not be of an ideal quality. And then the logistics around just this process in general, harvesting the cells from the patient using something like apheresis, transporting from the hospital to a processing facility. Then you're going to be processing the cells, which include a number of different steps. This whole processing timeline might take 19 or 20 days. There may be cold chain involved, so having to ship product back and forth cold or cryogenically frozen. You can imagine that this is all done in parallel with the patient's ongoing treatment program, so it's absolutely critical for the patient, and it's causing delays, and there's certainly some complications around that. There's the complications around the logistics of having to ship those back and forth in a timely manner, and then there's also certainly the product criticality complication, which is that if you're doing a patient-specific therapy, you're really only creating enough doses for that one patient. If something were to happen in transit, or if there were a contamination involved with the creation of those doses or something along those nature, that's all there is. And so you'd have to basically restart that timeline over again, and you can see how that could have potentially devastating effects for the patient. When we talk about these processes, the biggest differentiator between allogeneic, well, not necessarily allogeneic, between patient-specific and large-scale is that the scale of those operations really dictate how the facility is laid out and how it flows. There are almost completely different considerations taken for small individual scale production versus large scalable production. And then certainly process parameters can have large varying effects on that also. For example, if you are cryopreserving your cells and you're introducing DMSO as a cryopreservant, a lot of times there is a maximum amount of time before those cells have to be put into freeze to keep them from degrading too far. And so if you are doing a lot size of 1 liter or 2 liters, processing that through formulation and filling and putting into freeze has a dramatically different kind of time criticality than if you are trying to do that for a 1,000-liter bulk. Or cold chain, certainly, if you're just refrigerating, the cost implications of doing cold chain for a 1 or 2 liter batch versus a 1,000 or 2,000 liter batch can have dramatically different consequences for your facility. And then, as we'll go into later, there's typically very different equipment offerings in terms of processing equipment for the different scales, personalized scale versus large scale, and each of those carry their own implications as well. And so when I talk about large-scale production, I don't necessarily mean huge multi-thousand-liter systems. What I'm really referring to is multi-patient volumes. So it could be a 1,000 or a 2,000-liter size batch. It could also be much smaller than that. But the idea is that you are producing multiple patients' worth of doses at a time as opposed to a patient-specific dose. And again, this type of process really only applies to allogeneic cell production. And what we're finding is that this resembles much closer to a classic monoclonal antibody production in terms of upstream. Typically, your specific cell selection, activation, manipulation is all done prior to floor manufacturing so that you can set up a cell bank for consistency. And so then this looks a lot like what you would see in terms of upstream for a monoclonal antibody facility, where you have a cell bank, and then you are, thawing that working cell bank vial. You are going through cell expansion, and then there is a harvest step. Now, in this case, where the real difference is between, classic biotech, the cells themselves are the product. So when you are doing harvest, you have to take into account things like shear sensitivity plays a big part. And so there's lots of different kinds of equipment that can perform that activity. Centrifuge tends to be fairly popular, and there's a number of different offerings available on the market, both in terms of scale and ranges. And certainly you want to accommodate something that can have low shear and accommodate high cell densities for dosing and or further processing. And then this would be followed by a formulation step where you are potentially formulating into a cryopreservant buffer so that you can do long-term storage, because again, for an allogeneic product where you are having large bulk processing, it's advantageous to be able to store so that that product can be made in advance and available upon need. But then again, we discussed a little bit earlier that if you are formulating into something that contains DMSO, I know there are a number of cryopreservants available on the market that are DMSO free, but we're still seeing that DMSO is a popular selection. And so there are certainly considerations that have to be taken around that for processing times and getting your product into controlled rate freezing relatively quickly so that you can minimize degradation of the cells. After formulation, you'd be doing filling either into vials or bags typically, and then again a controlled rate freezing step likely so that you can then go into cryo storage for supply storage. And then for this type of production, you might have also an irradiation step where you might be cross-linking so that you don't have further cell expansion. This in some ways helps fix the cell count for a dosage, but what we are seeing is that it's much more in terms of protecting your product line, because if you have a generic cell therapy offering that can be administered to essentially any kind of patient, the last thing you want is for someone to be able to take those cells and then clone them and then have that exact same product line. And then when we talk about patient-specific production, which again is going to be autologous cell therapies or donor-matched allogeneic cell therapies, this is a much smaller scale because you're really only creating enough doses for that one patient. And so there's many more challenges associated with trying to do operations at a much smaller scale, but within a GMP setting. And again, because you might be getting cells harvested from the patient themselves, are the cells coming in fresh or are they coming in frozen? These are considerations that will need to be accounted for. Typically, in terms of processing steps, you'd have the cells coming in, you may have to thaw them. You'd do a fractionation and selection step. You'd activate your cells. Then you'd do expansion, harvest, formulation, and fill. And then if they're going out frozen, you'd be doing controlled rate freeze prior to shipping. And important to note that each patient stream for this kind of production is essentially treated as a separate product line, because if we're talking patient specific, right, we can't be producing any of this in bulk. So each separate patient stream is essentially its own product, and that has to be considered for things like facility segregation, material handling, things like that. And then the last complication for this type of production really is that historically equipment has not been as available offered in a closed processing automated solution. not nearly as much as is available for the the larger scale production that we were just talking about, allogeneic. So looking at where the regulations are before we get into how we lay out a space, I've got some quotes on the board here that I think are important to set the basis for where the regulatory understanding is in terms of how we can be doing production of multiple different product lines within the same space. And so when I say product lines, if we're talking allogeneic, this might be 2 or 3 different cell line offerings. If we're talking autologous, these would be separate patient streams. So manufacture of advanced therapy medicinal products in a multi-product facility is acceptable when appropriate risk mitigation measures commensurate with the risks are implemented to prevent mix-ups and cross-contamination. So that's laying the foundation that multi-product production is okay given that you are doing risk mitigation that's appropriate, which doesn't say much, but we'll get into more details as we continue through here. And then the second quote, the use of more than one closed isolator or other closed system in the same room at the same time is acceptable, provided that appropriate mitigation measures are taken to avoid cross-contamination or mix-up of materials. So for closed equipment production, this reads very similar to the industry's understanding for classic biotech, where closed process systems can operate in a shared production space. This, of course, assumes that there's measures in place to accommodate things like operational workflows, in the event that there's a spill from one product line, how that affects your other equipment, sample handling and mitigating mix-up of samples, things like that. And so then this further opens up the discussion of the other trends that we're seeing in classic biotech, so things like open ballroom concepts, potentially down-classifying your operational space in terms of air quality, more of the trends we've recently seen that help create more efficient, cost-effective operating facilities. But again, this is around closed processing equipment, and so you might ask, well, what if the process that we have, there just isn't a closed processing equipment offering available? Where do we go with that? So what about open processing? Here we see possibility of using more than one biosafety cabinet in the same room is only acceptable if effective technical and organizational measures are implemented to separate the activities, e.g. strict material and personnel flows defined, no crossing lines in the use of equipment in the same room. It is stressed that the simultaneous use of more than one biosafety cabinet entails additional risks, and therefore it should be demonstrated that the measures implemented are effective to avoid risks to the quality of the product and mix-ups. So this outlines that you can perform multi-product production in the same room using open operations within biosafety cabinets, but heavy emphasis on procedures in place to present, quote, mix-ups. And what they're talking about in terms of mix-ups here is really these operations tend to be more manual when you're using open equipment, a lot more operator-driven. And so they're really stressing that operations need to minimize as much as possible human error. Things like operators accidentally walking to the wrong workstation or accidentally bringing the wrong materials or leaving the wrong materials at the wrong workstation, because these patient-specific doses are so critical that any sort of mix-up like that could potentially have devastating effects on the patient. They're really just stressing that you can do it, but you need to really take careful consideration of how you lay out your facility and how you structure those operations so that you minimize those mix-ups as much as possible. The other flip to that is that closed automated systems, closed operation equipment systems tend to be easier to track and trace production in materials. And so, again, with manual operations, there's just that additional risk. And then to further emphasize your ability to do multi-product, what other kind of operations are acceptable? It is acceptable to conduct a manufacturing activity in a clean room which hosts an incubator which is used for a different batch or product if there is separated expulsion of exhausted air from the incubator. And then simultaneous incubation storage of different batches within the same incubator is only acceptable if they are physically separated, e.g. distinct cell cultures in closed vessels. When simultaneous incubation storage of different batches takes place as described above, the manufacturer should evaluate the possible risks and implement appropriate measures to avoid mix-ups of materials. So again, this just outlines that we can manipulate different patient batches within the room while other lots are in expansion stages in the incubator. You can also carry multiple patient batches within the same incubator given risk mitigation strategies. Again, heavy emphasis on preventing mix-ups, being able to track product lines through your production, helping ensure the operators aren't accidentally doing something that will cause quality issues with your process. The bottom line through all of this section, though, is that regulatory guidance supports the ability to perform multi-product or multi-patient operations within the same suite, within the same facility. Well, certainly within the same facility, but within the same operational suite, given that systems are closed or properly contained either within closed processing equipment or within local environment equipment like isolators or biosafety cabinets. And then certainly heavy emphasis on carefully planned procedures and that those are in place to ensure defined workflows and to prevent mix-up of materials or processes. So what does this mean for the processing space itself, the operational space? Really setting up a suite operational philosophy where you'd have various working bays so that you can be operating on multiple patient product lines within the same space. This maximizes that suite throughput and use profile and really helps decrease the downtime of your facility and maximizing the value that you are getting out of that infrastructure dollar. And while you might say that yeah, the regulations support it, but who's really doing that? We've worked with several well-established companies who operate in this manner with multiple product lines or patient lines within the same operational suite. So it is being performed right now. And again, this is really the approach to maximize the return potential of your cost investment. And when you think about autologous or donor-matched allogeneic therapies, this is really very critical from a scale perspective to increase your facility throughput, because if you're trying to do like one patient per suite, you're going to be either bottlenecking your facility or really limiting the throughput that your facility can handle. And so talking about site throughput, the way that we usually approach the design of your facility in terms of throughput and accommodating that throughput starts with defining the target number of prepared doses per year. And so when I say doses, for an allogeneic bulk process, this would match your annual production demands, right? You might have a forecast for how much product you think you need every year. This would be that amount. And then for autologous or donor-matched allogeneic therapies, this would be the number of patients per year that you're anticipating being able to accommodate. And then process schedule really is the cadence of your process, how long it takes front to back to be able to perform that operation. For autologous, that might be something like 19 to 20 days, for example. This really fixes how often that equipment can turn over when you are performing your operation, and then the scale of that equipment will determine how much product is made at that same process schedule cadence. So for allogeneic, that would be the scale of your processing depending on how large you get in your bulk process. Certainly for autologous or donor-matched allogeneic, the scale is really one patient worth per operation. And then again, that multi-product operational philosophy, how many different processes can you do within the same suite, is going to dictate how many suites you need to be able to accommodate your overall facility throughput. And so that, again, is a critical element to this. When we talk about cost considerations, there's a number of different costs, but there's also some more hidden costs that I'll try to help discuss a little bit while we're discussing here. So obviously, infrastructure cost is, your facility, the architecture, the building itself. That's a fairly obvious upfront cost, and that's going to be dictated a lot about the number and size of operational suites that are required, and then certainly all of the supporting areas to accommodate that operation. And with that, again, comes airlocking. I think a lot of people or organizations underappreciate how much space in your facility can be taken up with airlocks, especially if you are doing open operations. You might be operating in a grade B or an ISO 7 air quality space. There's a lot of airlocks that can be required to cascade the operator through your common corridors up to those operation spaces. And so you need to be cognizant that we're not taking up too much space with airlocks, because that's going to be costs that will be difficult to recoup. And then in terms of equipment capital cost, certainly there's processing equipment that's going to be based on the number of systems and the system type that you have. When we talk about open operations, and these are things like incubators and biosafety cabinets and centrifuges, not a very heavy cost there, versus closed automated systems tend to be a lot more expensive because they are a lot more complicated. But it's important to note that that one-to-one cost is not necessarily an equivalent representation, because the true cost of an open operation really should include things like the operational footprint. You might have a larger footprint required to do that operation because it's so manual. There might be a lot more utilities, certainly things around HVAC to accommodate higher room classifications, all those airlocks. Because the operations might be more manual, you will have a much higher labor cost typically. And then certainly there's the cost of the additional risk involved in doing manual operations versus something that's more automated. And then mechanical equipment, again, talked a little bit about that. HVAC air classifications can have a dramatic effect on the cost of your mechanical equipment. Cold chain requirements can have a dramatic effect cost-wise on your facility mechanical equipment, chillers and things like that. And then for operational costs, just touch on quickly, cost of goods, certainly there's the ongoing cost of components and disposable components. Certainly if you are offsetting your solution preparation and contracting medias and buffers, that's going to be an ongoing cost. And then a big one that needs to be considered is the impact of labor requirements for your operations. So not only is it going to be a manual operation versus an automated operation, how many people are involved in performing that operation, but it's important to consider how many simultaneous operations are going to be required for your facility throughput. So when we talk about large-scale allogeneic processing, that's a lot less emphasis on labor because it's essentially one or two batches that you're doing that will have a couple people involved, whereas if you are talking autologous cell therapy, patient-specific scale, where you need one or two operators per patient line, that's a lot more people involved if you're trying to treat 1,000 or 2,000 patients a year. So a much heavier labor investment there. And then there's, not listed here, but certainly facility operational costs, things like utility sampling, room viable monitoring, all of the labor that goes on, certainly quality and quality checks, all of the labor that goes on to support the operations on the whole. And so in terms of being cognizant of maximizing your facility value, there's a number of different things. One is certainly right-sizing your operational spaces, right? It's not, I think it's the wrong approach to think that automatically smaller is better because it's less expensive. Certainly you want to have an operational space that is appropriate for the operations that you're intending to perform in there. But being cognizant of airlocks certainly, and philosophies around if your facility must be unidirectional flow or bidirectional flow, certainly bidirectional flow is going to reduce the number of airlocks and reduce that cost, reduce that number of spaces that must be sampled. And then again, things like HVAC, utilities, labor, those all go into your operational facility cost. And so we talked about closed automated processing. I think this is really the key direction for this industry that equipment vendors are going to need to be taking a hard look at. Because we saw with the regulations, system closure is a key way to be able to do multi-product processing in the same room. As we've seen with classic biotech, it can, if you're removing things like biosafety cabinets, you can potentially operate in lower classified operational spaces, which is not only much cheaper to install, but much cheaper to operate. You have fewer airlocks, so it's a smaller facility footprint. You get much better utilization out of that facility footprint for your product lines, much better value for your dollar. And certainly if we talk about system automation, this is going to reduce things like the opportunity for human error introduction, could potentially provide better reproducibility and consistency in your product quality or your product output, resulting in more consistent product quality. And then certainly the more automated the solution, the reduced labor demands that you're going to need. Fewer operator interactions results in fewer operators required. And this in the end is going to result in a better quality product for a lower overall cost. And so I touched on earlier that there's not a ton of options available on the market for patient-specific scale, but vendors are noticing this, and they're starting to we're starting to see this shift in the marketplace. So things like the Miltenyi Clinimax Prodigy system, or the Octane Cocoon that was getting a lot of press, I think late last year. We're starting to see closed automated systems that can help support this patient-specific scale. Looking at the facility on the whole, on the left here is a list of, well, both columns here are a list of typical types of spaces or functions that you would see in a classic biotech facility. So not all of these are really needed for cell therapy. Some of them will have greater emphasis, some of them won't necessarily be needed at all. For example, if I'm highlighting some of the more impacted spaces, if you're doing an autologous cell therapy, you likely won't have cell banking whatsoever because you're relying on the cells coming in harvested from the patients. And then for those operations, are the cells going to be arriving fresh? Do you need to have spaces that can accommodate the logistics of accepting fresh cells and maybe storing prior to their being able to be processed? If we're doing an autologous or an allogeneic batch, now you likely will have cell banking or donor cell storage. And so those might be vast. It would depend on your process and how you store those cells prior to use. Process utilities can potentially be dramatically reduced for patient-specific scale. You can depending on how many patients per year you are trying to have for your facility, you can potentially get away with purchased water, or as we'll go into later, contracting out your solution prep entirely. And so you can dramatically reduce the number of process utilities that would typically be seen at a biotech facility. In terms of product storage, if your cells are cryogenically frozen or if they are fresh going out, do you need refrigerated storage? Is it going to be shipped just in time? Do you need, for example, an allogeneic larger scale production, do you need banks of cryogenic storage freezers to accommodate your annual productions or things like that? Certainly that needs to be considered is the cost of liquid nitrogen tubing. Piping tends to be vacuum jacketed, and it's very expensive to install, somewhere in the range of maybe $1,000 a foot. So if you're putting in a large bank of cryogenic freezers or controlled rate freezers, you need to be cognizant of how those freezers are laid out. You'd want to keep them local and preferentially close to your liquid nitrogen bulk system so you can minimize those piping runs as much as possible. Again, weigh and dispense and solution prep might be very minimal for a patient-specific scale, and so maybe you are purchasing media, maybe you are contracting out solution preparation for those buffers as well. That can be a way to remove weigh and dispense operations entirely from your facility, solution prep entirely from your facility, which can also potentially free up certain spaces within your warehouse and distribution as well. And then what we are starting to see is on single-use facilities, it's becoming a little bit of a trend for if all of your equipment truly is single-use disposable, that the need for equipment wash and autoclave systems, so things like GMP washers, GMP autoclaves, which are fairly substantial systems with fairly decent lead times and are critical to lining out in your facility in terms of an infrastructure because they are typically installed in the walls and mounted to the floors. Those systems are now being reduced out of facilities. So if you have a fully disposable process train, there really isn't a need for a washer or an autoclave. And so if you can remove those from your facility, now you can further reduce process utilities as well as removing those spaces that are typically needed to accommodate a cleaning wash operation or an autoclave operation. And then, again, while not specifically emphasized here in terms of a highlight, HVAC is directly impacted by the air classifications that you choose for your operational space. So looking real high level at a case study where we can look at comparative costs between a couple of different design considerations. This first one that we'll look at will be kind of dialed to 1 in terms of flexible, scalable, really the most conservative option just as a comparative note. Single patient per suite performed in an open manner inside of a biosafety cabinet. So you've got a grade A biosafety cabinet with a grade B, or an ISO 5 biosafety cabinet with an ISO 7 background, and then cascading airlocks from a common supply corridor. Here it's shown bidirectional, and then with a process cycle time of somewhere in the range of 19 to 20 days potentially, and accommodating things like your process setup, your operations itself, and then your cleanup. You might only get roughly 18 patients a year coming through this suite. So you can tell it is not very cost effective. The cost that I have listed there is what I refer to as an infrastructure efficiency cost. So it's sort of the cost of this space per patient per year, so dependent on the throughput of the suite itself. And the cost for this is really based off of a dollar per square foot, based on the air class and the type of equipment. This does not this number does not really capture the operational costs. It's really more of just the install cost. And so if we, with only one biosafety cabinet, if we maybe take advantage of the fact that you could have multiple patients in incubators, you might be able to flip that biosafety cabinet in the middle of the day and do one patient operations in the morning and then one patient operations in the evening, but really you're probably only going to be able to get a throughput of 2 patients per day through this. And so maybe you double your annual patients per year, but it's really still going to be not a very cost-effective solution. So if you're trying to do 1,000 patients a year, this philosophy is almost a no-go in terms of trying to design a facility that can do large-scale autologous production. And so then we look at a much more flexible option in terms of multiple patients per suite performed in an open manner in biosafety cabinets. So again, segregated operations, segregated workflows, but concurrent production, and this gives you much better efficiency for space. The patient range that I have listed there is going to be incorporating multiple patients per day in the biosafety cabinet. So that's why you might have, if you're going to do 1 per day versus potentially 2 per day, you can get up much higher. And while the facility footprint is slightly larger, you're getting a much lower cost per patient because the space is much better utilized. Again, this does not really incorporate operational costs or labor costs. So it's still talking about open manual operations, so it's going to be things like how many personnel do you need to perform this operation, the sampling for viables in the environment through the airlocks in the space itself, and then certainly the ongoing HVAC cost of that higher air classification space. And then the last one is going to be multiple patients per suite, but performed in closed systems as opposed to open systems. So in this example, we've declassified the room from a grade B background to a grade C background, or going from ISO 7 down to an ISO 8. And it's got the operational space itself has a much smaller footprint, since the closed systems typically have onboard analytical devices, they don't require quite as much additional equipment to support, so the space itself can get smaller to accommodate those operations. And then because the room air quality is declassified a little bit, there's fewer airlocks required to gown into that space. So the cost listed here is not necessarily the lowest cost per patient. I think that's because we're considering that there's only 3 systems within that room, and so because you might not have onboard, or you might, we're considering that you're doing expansion within those systems, so you're limited in that sense to how many patients per year you can go through. You can't double up on like you could with the biosafety cabinets. But there's certainly ways that you can maybe potentially get around that to maximize the use of those systems and still maintain a closed environment, and so then you can pull your infrastructure efficiency costs even further down. And certainly with the grade C or ISO 7 space or ISO 8 space, there's other aspects to consider, things like lost time gowning, trying to gown into the higher classified space, reduced labor for the automated processes versus manual processing, the sampling regime for all the environmental viables, sterile material logistics. So if you are bringing materials into an ISO 7 or a grade B background, it's anticipated that all of those materials are sterile, whereas for a grade C or ISO 8, that is a relaxed requirement. And then certainly HVAC mechanical operational costs. And then this case study is also maybe a little skewed because you might not be doing all of your operations within the same suite the way we have shown it here in these examples. You might be doing, for example, cell washing, selection, activation in one suite, and then offsetting expansion to a lower class separate suite, and then maybe you're doing harvest and formulation in one suite, and then filling and cryogenic freezing in a 4th suite, for example. But I think the concept of throughput per cost still holds pretty true in terms of trying to effectively manage your facility efficiency. And I think looking at this case study example right here, I think what we're seeing from a cost per patient per year, really the key design element would be to utilize closed systems with a number of the operation steps in terms of washing, selection, activation, and then where the time-heavy step is, which is expansion, you could offset that operation into a separate closed system like a wave bag or something like that, which is the longest time step. And so then you could be really maximizing your throughput through these closed systems. You get multiple patients a day on your closed systems potentially, and really maximize that value for your facility then. So that's all that I have for today in terms of content. I hope this was helpful and that everyone was able to take something away from it. And I like to open the floor to answering some questions now. Thank you.
Okay, thank you very much for that wonderful presentation, Peter. And with that, we're now ready to start the live Q&A session. So please submit any questions you might have using the Ask a Question box on your screens, and we'll try and get through as many as we can in the allocated time. So, Peter, the first question then, are you seeing a trend in the industry heading more to either patient-specific or off-the-shelf?
Yeah, good question. So From industry polls that I've gotten a chance to take a look at, the trend in thinking seems to be heading towards allogeneic in terms of being a potentially better market offering. I think this is largely based in part to the ease of production logistics and manufacturing operations. But I think, not performing it myself, I imagine finding a cell line that's both effective and widely accepted enough is probably pretty difficult, and this is reflected in the fact that we don't really see we see very few true allogeneic offerings in development right now. So while I think people believe that allogeneic might eventually be the way to go, I think it's going to be a hurdle to actually get those cell lines developed.
Okay, great. So the next question then, carrying multiple cell cultures within the same incubator seems very high risk, but are you saying this is allowed?
Well, I'm just relaying what the guidance documents are saying is acceptable. So I think it's important to note that, as with pretty much all the guidance document statements, the real onus of proof is up to the company to show that their manufacturing methods are, safe and proven and reliable and consistent, and ultimately that they're bound in quality. So while the guidance documents say that you can do it, they also say, a lot of times that you've got procedures in place to prevent mix-ups and to ensure quality and things like that. So it'd be up to the operating company to really show that their methods pose minimal risk to the patient, and certainly things like how you reduce those chances of mix-ups and how you ensure quality, all that's going to be scrutinized.
Okay, now we're actually getting a lot of questions in, but we are going to run out of time soon, so this might be the last one. If you have 2 patients per day per BSC, why do you show the patients per year so low?
Yeah, this is a fair question too. I think this is the numbers that I was showing are a little biased. I maybe didn't explain it very well, so I'll go into it in some more detail a little bit. For the 1 patient per suite, I think it's a pretty clear breakdown. You've got, maybe 340 days of operation, and if you have a 18 to 20 day turn per patient, you're getting, 18 to 19 patients per year through that suite. For the multiple patients per biosafety cabinet with multiple biosafety cabinets, the numbers can probably be much higher. I think the case study I was showing is maybe a little unfair because if your design philosophy is to do multiple BSCs with multiple patients, you wouldn't design a facility to just do every operation in one suite. You'd frame it in a way. But I guess I was framing the study in a way to try to compare like one space to another to help show the benefits of closed system processing and also just show apples to apples. I think if you were going to do a multiple biosafety cabinet with multiple patients, you'd be limited by the incubator space. So in theory, if you're running 3 BSCs with 2 patients a day per BSC, you could push through something like 2,000 patients a year. But the suite would be seeing very heavy traffic, which would introduce its own risks. You've got the more operations you're doing, the more chances of mix-ups and things like that. And then at that point, you'd really need additional space for all the incubators and then the further downstream operations. And so now you've got additional rooms to accommodate those, and the model becomes very different and difficult to compare against like the 1 patient per suite mode. So that's why I was kind of trying to show it like that. And then that kind of also applies to the closed systems. I think if you were going to do closed systems, you'd look at the bottlenecks, which are really like the incubation expansion period. And so you'd try to offset that operation maybe to additional pieces of equipment like a wave reactor or closed containers and incubators. And so now you can really increase the throughput through your closed systems and have multiples of your expansion equipment. And so you could do all that in one suite and reduce your airlocks, or maybe there's strategic reasons to segregate those into separate rooms also. But certainly the nice thing about closed systems and doing all of your operations in one closed system, like how it's being shown, is that it's more automation control and less operator labor, and so it's probably going to be the lowest operating cost overall, but definitely is going to maybe bottleneck your throughput a little bit.
Well, thank you very much for that answer, Peter, and thank you for answering all of those questions. That's all we have time for today, but if your question hasn't been answered live here, and I know there are quite a few, then don't worry, because we will pass all of the questions over to Peter so that he can follow up. And if you wish to revisit the topics covered today, then this webinar will be available on demand in the next couple of days. So thank you again to our presenter Peter for sharing his insight, and thank you to everyone for attending. I look forward to seeing you at future webinars.